Hello, everyone. Welcome to another episode of Nerd RX podcast. I'm your host, Barkha, and today we have Dr. Deepika Jayaprakash, and she is going to talk about fluorescent competition assay. And thank you so much, Deepika, for being with us here. Welcome to the show.
Hi, Varkha. It's my pleasure to be here, and thanks for the invite.
It's my pleasure as well. So let's talk about fluorescent competition assay. And before we get into the topic, would you please introduce yourself to our listeners and so that we just get an idea what is your background and where you come from and what are you doing currently?
For sure, yeah. So, as you all know, my name already, I'm Deepika Jayaprakash. I'm from India. I did my bachelor's in dentistry from India, following which I did my master's in periodontics. Since it is a clinical subject, I got into practice, both consultations as well as in a clinic where I was working both as a general dentist and a periodontist. At the same time, I also worked as an academician where I would train dentists and periodontists. So this rich clinical experience actually got interested me into PhD program. Particularly, what I was intrigued was with these pre-malignant lesions that I would often come across in my clinical practice. So these pre-malignant lesions are these white or red, looking mucosa in the oral cavity, which has a tendency to become cancerous in future. Although there is immense studies in the literature which tells us when to intervene, how to test, which ones are at higher risk for developing into a full-blown cancer. We still are not sure as to how or what are the processes that are involved in the progression or sustenance of this precancerous lesion and subsequently a conversion of them to a fully blown cancer. So that's what made me interested in this field of oral cancer, particularly the oral squamous cell carcinoma. And with an interest in pursuing my PhD studies in this, I applied to PhD program, and I was very fortunate to be in to get admission in UNC because it's a very different program from others, where the program allows us to take up PhD mentor outside of the dental school, unlike other schools.
Okay.
I could take it in a medical school. I could take it in Duke or NC State, wherever I have an interest in. So that
Okay.
was really nice. So that's how I got into Waziri Lab, where my mentor is working on DNA damage pathways, in particularly in cancers, in the progression as well as in drug resistance in cancers. And he was quite interested when I said I want to work in oral cancer. And so I ended up having a project under a broad umbrella of head and neck squamous cell carcinoma.
Okay. Wow.
So
That's a quite transition from being a dentist and now into hardcore research scientist.
that is true. That is true.
Wow. Thank you so much for that wonderful introduction and congratulations on all your accomplishments, by the way.
Thank you.
So, why did you pick fluorescent uh, competition assay for this episode?
Yeah, I particularly got interested in this one because of my thesis topic. Like, my thesis involved a CRISPR-Cas9 screen, which I did for around 3 months.
Hmm.
Following that, I need to validate the screen. Now, my screen involved, of course, as the name suggests, CRISPR-Cas9, I will be knocking out certain genes. So my screen involved a library of single guide RNAs that would delete most of the genes that are there in the human genome which are involved in the DNA replication and repair pathway. So I delete them and I continue carrying the cells for a period of a month or so for each replicate, sorry, 1.5 months or so for each replicate. And I studied the effect of a particular gene of interest for me. I turn on the gene and I see what is the effect of that gene on the depletion of these DNA repair pathways. Now we got the results, we got it analyzed with the help of a collaborator, and the next part was to understand whether or not these hits that I've got from the screen are actual hits. Now, in order to
Correct.
validate them, I got interested in this particular assay called fluorescent competition assay.
Okay. Okay. So, just for our listeners, what is fluorescent competition assay?
Yes, simply said, this is a competitive growth assay.
Okay.
Basically, we are trying to test what is the difference in proliferation rate
Mm-hmm.
between the samples of our interest. Like whether we delete a gene or we use any inhibitors or drugs, we want to test what is the proliferation or growth rate of these cells when compared to the non-deleted or when compared to ones without the inhibitors or drug. So, to compare these things, we use fluorescent competition assay. And this assay could be expanded to other applications as well. But currently, it's mostly used to test the effects of a particular gene deletion in a system,
Mm-hmm.
or test the effect of any inhibitors or drugs that you're using in a system.
Okay. So what steps are actually involved in the assay? Like, how long? Could you give us an overview of how long from start to end of this assay takes, and what are the steps involved basically?
Yes. I got interested in this particular assay because it can be chronic. You can extend it to a considerable length of time. And I wanted
Mm-hmm.
this because this mimics my screen. My screen took about 1.5 months for each replicate, and I wanted to replicate the same conditions for my validation so that I'm sure that these genes are really the hits from my screen, which I can take for downstream pathway analysis. So that's
Mm-hmm.
one thing. It can be short or as long as you want until the cells die, basically,
Yeah.
depending on what, of course, you're using. Because I wasn't using inhibitors. If you're using inhibitors or drugs, maybe your assay will not be that long. Since I was interested in testing the effect of a gene induction, mine I
Yeah.
could take the validations long too.
Mm-hmm.
So in this assay involves mixing colored cells, that is fluorescent cells, together
Mm-hmm.
and growing them for a period of time. So what I mean by that is first you have to generate a colored cell or a fluorescent cell. Now you can do it in different ways. You're testing your gene deletion, right? So you have one cell which has your gene deletion there. The gene that you delete there is the hit that you have got from your screen.
Correct.
So you can either generate a colored cell from this transduced cell, or you can generate a fluorescent cell from a cell which doesn't have this gene knockout, which is on its own. So I chose to get a fluorescent or colored cell from a cell that doesn't have this deletion of the gene, so that I keep the other one constant or
Okay.
non-colored, and I mix both the cells and culture them over a period of time. So what I do during the process is, in the beginning, when I'm seeding the cells in this mixture, I analyze them by flow cytometry, which means that I get a visual of which cells are green and which cells are not. I get a nice ratio of which cells are green or which cells are not with this analysis.
Okay. Okay.
And once I seed them, I culture them for a few days, and I keep whenever I reseed them, for, say for every 3 or 4 days, I reanalyze them by flow cytometry to see the difference in ratio of the cells that I'm culturing. So that ultimately gives me the percentage of colored and non-colored cells which are changing over time as a result of this gene deletion, as well as my interested gene uh, expression.
Okay.
And we would have certain conditions to normalize, of course, like for any assay, that you would have some normalization factors. I would have some non-targeting gene that I would introduce as a single guide RNA that will not have an effect on any of the genes. That means it doesn't delete any gene. That will be one of my control. And another control, or rather the normalization factor that I use here, is basically the percentage of cells that I have seeded on the day zero.
Hmm.
So that will help me understand what is the difference in percentages of the cell as the cells grow with time.
Correct. So you mentioned you are using cell lines, right? Can you
Right.
just tell me what kind of cells are you using for this?
Yes. So I'm using a preneoplastic cell line from the oral cancer.
Okay.
So yeah, so this is the cell line, and I'm inducing a gene of my interest in them. So I have control conditions where I do not have any gene of interest, and and my test condition where I am inducing the gene of my interest in these oral preneoplastic cells.
So these cells line are like, do you buy them?
Yes, they're commercially available. That's right. They were
Okay.
developed from a region around the cancer in the oral cavity, which
Okay.
have the preneoplastic features
Uh-huh.
histologically. So
Hmm.
those were isolated, and the cell line was developed.
Okay. Okay. And okay. So you don't have to like, do you grow the cell lines from a new commercially available like every time, or you can like keep reusing the cell lines for future experiments?
Oh, yes, these were spontaneously immortalized. So, which means that I can keep growing them. Yeah, I can preserve the cells, and I can keep taking a fresh vial each time I want to conduct an experiment from the big batch of cryopreserved cells.
Okay. Okay. And how long do you need to grow the cells?
For the screen itself, that took like 1.5 months. But before the
Okay.
beginning of the screen, I had to generate these cell lines that express the gene of my interest in a conditional
Hmm.
manner. So that took me like 15-20 days to generate and select those cell lines for that purpose.
Okay. Yeah. I remember in my PhD, I used cell lines to test my drugs, but those were like hex cell lines. And that
Mm.
was like, you just seed them, and day 5, you can run your experiment. So that used to be
Ah,
like really short and sweet as compared to like months
yes.
of what you're talking about.
Yeah,
Mhm,
yeah, that's
interesting.
right.
So, are there any alternative techniques we can use instead of fluorescent competition assay to get the same results what you're looking for?
Yes. So this, as I mentioned, it's just a growth rate or proliferation rate assay. So any assay that will determine the growth rate or proliferation rate could be used. It could be as simple as counting the cells by
Okay.
measuring the population doubling over time,
Uh huh.
or it could be more quantitative, like we can use calorimetric assays like MTT, or even the cell titer glow assay, which is based on ATP generation. Again, it's a luminescent ATP assay. So any of these or proliferation assays can be used as alternatives. But that said, this one that I'm using has specific advantages. One major reason that I'm using it is for the time that I can use it for. Like MTT and cell titer glow are like short assays. You do it acutely. But since I wanted to
Mm-hmm.
reproduce the conditions of my screen and do it for about a month at least, I wanted to choose a GFP competition assay. That is one of the main reason. And a couple of other advantages that the system has is that it has an internal control. Like I said, we seed mixed population. It's not like
Mm-hmm.
there is a different population from which we compare and normalize. Although normalization still occurs with a different set, like a day zero seeding or whatever, but still there it is internally controlled because it is a mixed population that you are seeding. And also, like some other acute assays that I mentioned, like MTT, this is quantitative and it's visual. That is also a very good advantage of this assay.
Mm-hmm.
Um, and one other major advantage I see is it reduces the conditions. Although when you go to see, it's not much, but when you compare it with the assays like clonogenic assay, which is something that we also routinely employ in our lab, where we see the cells in low densities. and we will culture them over a period of time, say 7 to 10 days, to look at what the effects the inhibitor or the gene or the gene deletion depletion would have had on these cells. What happens is, since the clonogenic assay, you will have multiple controls. You're con- uh, controlling it for the seeding density, controlling it for the gene, controlling. So that's one of
Okay.
the huge advantage I see here. And another major advantage also is that it is compatible with high throughput screening. Like since we are utilizing flow cytometry, that will have accommodation for a plate. Like you could easily do it in a 96 or a 384 well plate format that easily works for this.
Okay. Great. And what about the other side? What are the disadvantages of these techniques?
Yeah, the advantage itself is a disadvantage that it's time consuming. Yeah, it takes a long
Right.
time. That said, one other thing is that you require fax accessibility. You should have a flow cytometry either in your lab, or you should have accessibility to core, which then means you will have to keep going to the core back and forth to get the samples there. As this has to
Hmm.
be run live, it'll be time sensitive. You will have
Okay.
to do it immediately. Other than that, the technique itself, like since we are testing the effect of the gene, the CRISPR-Cas9 technology itself, so all the disadvantages that come with the CRISPR-Cas9 also applies here, like whether there is a complete gene deletion or not, or whether the deletion is, even if it's partial, if it has had an aberrant protein that has been generated, what is the effect of that is unknown. So that could hamper the results that we get. So it's not a foolproof kind of assay like any other assay. You have to
Yeah.
couple it with other assays to have
Hmm.
a complete idea of 100% surety of whether the results
Hmm.
are accurate or not.
So, is there a way you can determine if the gene you are interested in is either completely deleted or partially deleted? Like, can you find that out?
Yes, so we routinely use the protein expression experiment, like we do Western blots to do that.
Okay.
But that is, of course, limited by whether you have a good antibody for that particular
Correct.
protein.
Hmm.
Um, Other way of doing it, which we are also considering, is doing a tide assay,
Hmm.
where we collect the DNA from the cells and we sequence them. And the sequencing data we put on this website, where it quantifies the insertions and deletions that the sequence gives when compared to your control sequence that you input, which will help us understand whether or not we have good deletion of the gene or not. But that too is not foolproof. So we can do a combination of both protein and tide assay to be sure whether or not the gene is deleted in this case. So that would be
Hmm.
more helpful.
So, what's the threshold? Like, if it's not 100%, what percent would be okay for you in terms of deletion?
I think an 80 to 70 to 80% also would work, as long
Okay.
as you are seeing a phenotype. Like you do see that there is a change, that you're able to validate your hits from the screen.
Hmm.
And you see a gene deletion of about 70 to 80%, I think I would still go with the result.
Okay. Okay.
Yeah.
I think in certain assays you would, for example, if you use like an agonist antagonist, I think there, I'm assuming you would want almost everything to be deleted. Otherwise, it will still give you um, a response if it's not completely deleted. So I think it depends on what you're looking for. It's like
Mm.
experiment based.
Which is true, which is true. So which is why, as I mentioned, complementing this assay with others would be a very good idea.
Yeah.
Like for instance, in my particular results that I got, I had inhibitors for two of the genes that I saw. So
Mm-hmm.
I started using inhibitors to and doing clonogenic assay to complement the
Yeah.
fluorescent competition assay. And also I did even short-lived assays like using siRNAs to deplete the expression of the mRNA. That also could be done, which will be acute, which will not have the effect of the knockout, which will at least suppress the expression for at least 4 to 5 days. That also could be used as to complement the fluorescent competition assay.
Yeah. So, like, no experiment is foolproof. You always need a backup to
Right,
prove your
right.
point.
Yes, yes. So true.
So, in terms of expertise, how easy is it to learn? Is it like really complicated or a complete novice? Do you think they can pick this technique up quickly?
Actually, it is pretty simple as long as you kind of know certain techniques. Cell culturing, of course,
Mm-hmm.
um, that you would, I mean, that's the basic, so it is very easy. Then you
Mm-hmm.
would have to know handling the flow cytometer, which is not very complicated because you're not sorting the cells or anything. What you're
Mm-hmm.
doing is just running them, setting some gates for the particular fluorescent marker that the emission spectrum of the fluorescent marker that you're using. That is not too bad. You can just set up one pilot assay and you're good to go with that.
Mm-hmm.
And if you're actually giving it to the flow core, you're even better. You just need to culture the cells and run to the core to give it to them to run the
Okay.
assay for you.
Mm-hmm.
So it's pretty simple, I would say. And of course, you should have generated the fluorescent cells. That calls for a little experience with generating the virus for getting the cell lines done and infecting your cell lines with the virus to and selecting them. And if sometimes what happens, mine I use the population of the cells that I got from this fluorescent cells that I generated. But my colleague could not do that because the intensity of fluorescence was different. Each cell was giving different fluorescent signal, which is why she ended up getting a single clone, isolated a single clone, and from that she did the fluorescent competition assay. So this
Mm-hmm.
calls for a little experience which can be easily gained. It's not that difficult, but with that
Mm-hmm.
experience, I think it would be pretty
Mm-hmm.
simple to handle this experiment.
Okay, and I think from my experience, like I've been trying to label neurons, and we were following like what our collaborators are doing. We got the labels from them, but somehow it would not work for us. I think it's the dilution issue. So
Hmm.
again, it's like really, I feel very experiment specific. Like I was trying to label like prefrontal cortex neurons, they were doing hippocampus neurons. So I think there is something going on there. We were not able to do that. So,
Oh, yeah, yeah. So it's so different when you work with the same system, still it is
mhm,
so different, right? In the
exactly.
same field of study. Oh, my, yeah, that's
Hmm.
true.
Hmm. And figuring out the dilution, I think, is another problem for us.
Hmm.
So, hopefully, it
Hmm.
works out in the future.
Yeah.
So, let's move on to the costs involved here.
Uh, Considering the other expensive assays, I think the cost involved is not too high.
Okay.
Now, of course, I followed it up after the screen. So, synthesis of the screen and all had a lot of cost because we used a library of single guide RNAs. So we had to buy pools of RNA, single guide RNAs. For each gene, we had like 10, and we had some non targeting guides. So, all that cost a lot of money, and generating that pool of library also cost money. But as such, this particular assay, I think it is not that expensive as long as you have access to flow cytometer. We have an in-house flow
Okay.
cytometer, which is a very basic one, not a sorter, but
Hmm.
just the basic one. So that helps. Or like, I mean, if you're from some other lab, you're welcome to use in our lab. So that way also collaboration will also help, because I
Yep.
know many labs in our building come to our lab to do the basic flow cytometry from our lab. So if
Hmm.
that can be done, so there also there can be cost cutting, and it would essentially be generating these cell lines, the fluorescent cell lines, and doing it. So the cost, I would say, is not that huge, considering the other assays. Yeah.
Okay, that's good. So if the cost is not so much, it means that many people can develop this technique and use like routinely in their labs.
Yes. Yes.
Because there are certain techniques we covered in the previous episodes are like just not feasible for everyone to do in their lab,
Mm.
like high throughput screening and cryo electron microscopy. Like from what I've heard, like they usually pool a bunch of different departments or even institutions and have like one cryo EM between them. Because
Oh
so
yeah.
in
Yeah.
terms of that, this is really nice change.
Yes, it's a nice change. Yeah, I heard that high throughput. The I think the screening, the compound screening episode of yours. Yeah, that was
Hmm.
very fascinating. Yeah, these
Hmm.
high throughut assays are always very fascinating and costly or expensive, I
Costly.
would say.
Hmm.
Yes.
Hmm. Awesome. Thank you, Deepika, for all the information. And just for our listeners, would you mind recommending any article, interesting protocol you have so that I can link that down in the description for the future references?
Yes, certainly. There is this fascinating research article that was published in 2021 by Hundley et al. In Nature journal. So this does a CRISPR-Cas9 screening just like us, and it's a huge screen that they have accomplished, which is very fascinating. And they go on to validate that by GFP or fluorescent competition assays, which is something actually inspired us to take up for validation of our screens as well.
Oh, wow.
Yeah, I would definitely recommend this one, a huge screen of ubiquitin pathway. Yeah.
Awesome. I will make sure to have that linked down in the description for our listeners. And once again, thank you, Deepika, for giving us your time and discussing fluorescent competition assay.
Thank you. It was a pleasure.
And for listeners, please stay tuned next week for another episode. And if you guys have any suggestions for the topics I should cover, please email me at barkha@nerdrxpodcast.com. And remember, it's good to be a nerd. Bye.